one type of globular protein is an enzyme. And so we're going to spend some time discussing how the enzymes work. So for many of the thousands of reactions that we have in the cell, energy must be put in for the reactions to occur. What is that energy called? Even for a spontaneous reaction, you have to put some energy in for it to go, like to get that ball rolling. Activation energy, that's right, it's activation energy. So in the lab, what we can do to put that activation energy, what can we do in the lab? You can heat it, you can put a little flame underneath. Can't do that in the body, right? So yeah, that would not work. So what do you have to do? You have to use an enzyme to lower that activation energy. And that energy you can think of it as taking the place of that heat in the lab. So we're going to talk about catalysts. We're going to do a little review of general chemistry. We're going to classify them, and then we're going to go on to the model of enzyme kinetics called Michaelis-Menten. So for non-allosteric or single subunit enzymes, it's modeled by Michaelis-Menten model. So we're going to go through that mathematically. For the allosteric enzymes, we're not going to go through it mathematically. We'll go through it qualitatively in the next chapter. So to make the Michaelis-Menten mathematics a little bit easier, we make it linear, and we use a plot, and that helps us understand inhibition. And so let's talk about a review of general chemistry. All right, so this is our little plot of a energy diagram of a reaction. So we know that this is a spontaneous reaction because the reactants had more energy than the products. So the delta G, change in free energy, is going to be negative because remember that triangle means final minus initial. So final is a small number, minus initial is a big number. Small number minus big number gives a negative number. So when you go down in energy, it gives you a negative change in free energy. That's why a negative delta G means a spontaneous reaction because you're going from a big number, more energy in the molecule. So let's talk about free energy. Free energy is the energy within a molecule. Remember I said angry molecules. This isn't, some of them are going to be, very tense. They're going to have, I talked about the phosphates next to each other being angry, a lot of energy in them. So those molecules that have more energy within the structure have a higher free energy. So if the reactants have more energy within the molecule than the products that are more relaxed, then that's going to be a spontaneous reaction. But here, even though this is a spontaneous reaction, it needs some energy to go. And that's the delta G of activation. And so that is where you have to put that in order to get to that spontaneous reaction. Some molecules will be able to get there through the temperature. Do you guys remember when you were in general chemistry and you were talking about the kinetic energy of molecules and how the temperature, like say the room in this temperature is 74, right? But not every molecule has that kinetic energy that corresponds to a temperature of 74. There's a couple that are very, very low kinetic energy, and there's a few that have very, very high kinetic energy. It's a distribution. So some of the molecules have enough free energy, kinetic energy to get over this. Not all of them. So it was still, even at room temperature, some will go forward. But to get all of them over, we have to put in the energy to get over that transition state. So what the enzyme does is it makes this hump a little bit smaller, or a lot smaller, so that more molecules can get over that hump. So kinetics tells us about the rate, and the rate is dependent upon how many molecules can get over that transition state. Whether it's spontaneous or not is a state function. It only depends on whether on the beginning and the ending conditions. So there's another thing here called the transition state. And the transition state is the point of highest energy, and it's halfway between the reactants and products. And we'll talk more about what a transition state is. And Michael Jackson had this one video called Black or White. And he had, did anybody ever see that one? You've seen that one? Okay. So what it is a bunch of phases, and it just transitions from one phase to another. And the way I think about transition state is, let's stop it right in the middle of the transition from one phase to another. Does it even look human? If you're in the middle of two phases, it's pretty weird looking, right? It doesn't really look like the reactant, doesn't really look like a product. It's this weird thing. And that's what a transition state is, this weird thing halfway in between two phases. That's the way I see it. And next time you see that video, you'll think of transition states. All right, so this is what our catalyzed reaction can do. It can lower that energy of activation. So it doesn't change whether a reaction is spontaneous or not. It's not going to make a non-spontaneous reaction spontaneous. It's only going to change the rate of that reaction. Okay? So the change in free energy, change in enthalpy of the reaction is, remember Hess's law, products minus reactants. It's also not going to change, because it doesn't change the free energy of the reaction, the enthalpy of the reaction, it's not going to change the equilibrium constant, right? Because the equilibrium constant is dependent upon the change in free energy, the delta G. Now, it can change the free energy of activation quite a bit, all right? It can change, it can increase the rate of the reaction by 10 to the 20th. Let's take this example of breaking down hydrogen peroxide. I like to do this with the kids. I call it elephant toothpaste. If you take new hydrogen peroxide and you put it in your shelf for 5 years, it might not be good anymore because it breaks down to water and oxygen, right? You don't use old hydrogen peroxide. Although the bottle I have at home doesn't have an expiration date, it really should, because it's not going to be effective. Because this is a very high energy of activation. It takes a very long time for all the reactant molecules to go to products. So we'll call the relative rate of the uncatalyzed reaction 1. But if you put catalase in here, if you put catalase in with hydrogen peroxide, it breaks down very quickly. It can do it in seconds. And catalase, you can find it in yeast. You can release it by making yeast. So what I have kids do is I take a graduated cylinder, a big graduated cylinder, and I put some soap in with hydrogen peroxide. And then you guys can do it at your next party. Then I take a little measuring cup and I put a package of rapid rise yeast and put it with some hot water and mix it up. And I have a kid pour a little bit of that because it will put some catalase in with the hydrogen peroxide. And immediately it starts to catalyze this reaction here. And this is what state is this? Solid, liquid, or gas? Oxygen. It's a gas, so it's going to fill the soap bubbles, and it will just start spewing soap bubbles all over the place. It's like lava. You know, it's kind of neat. It's a lot of fun. You can use a platinum surface in a lab, right? And it will catalyze it, but it only catalyze it about half as fast as catalase does. So it really does it very quickly. Oh, the fun that I have with the kids. I have 1,000. If you have a party coming up, or if you have any kids in your life, I have a million kitchen chemistry things that I've worked through. It's a lot of fun. So we don't have platinum in our body, but we do have catalase, and it's going to break down hydrogen peroxide. So an enzyme is a biological catalyst. Okay, we all know that it's a protein. Now, how does that protein act as an enzyme? How does it do that? What it's going to do is it's going to take that reactant and make it look like the transition state. That's how an enzyme works. It takes in the reactant. We're going to call that reactant a substrate. So a reactant is called the substrate. So this word I'm going to use from now on, instead of reactant, like the hydrogen peroxide, H2O2, is the reactant in that one reaction. We're going to call it a substrate. And we're going to make an enzyme substrate complex. And that enzyme substrate complex, that enzyme is going to take that reactant and change it so that it looks more like the transition state. And it's going to change that energy of activation. Now, we mentioned that if we increase the temperature, we can make a reaction faster. But will that work with our enzymes? Oh, it's true. You increase temperature, all right? If we're at 0 degrees Celsius, an enzyme isn't really going to work that well. And we increase temperature, it's going to increase the rate. Why does increasing temperature increase rate? More collisions, is that what you said? More collisions. So in order to have a reaction to happen, you have to have a collision between the two reactants, right? Something has to react, has to bump into something else in order for it to react. I mean, the hydrogen peroxide has to break down, but in most cases when we have two reactants coming in, we have to have a collision between those two molecules in order for that to happen. So if you have a higher kinetic energy with a higher temperature, you're going to have more collisions. More collisions, more chances for the reaction to happen. Now, I'd like you to change this a little bit. I want you to change this plot a little bit. So here we have activity and temperature. I want you to go up, and then I want you to take this and draw a straight line down instead. Why would I draw a straight line down? What's going to happen once the protein's going to denature, right? You're going to start breaking apart those intermolecular forces and cause the protein to denature and no longer be able to catalyze the reaction. So temperature is one effect. Now, also, because proteins enzymes are proteins, the pH must not be changed drastically. But it doesn't have to be pH 7 for an enzyme to be active. Let's take a look at this. All right, amylase that's in your mouth, it has an optimum, this is enzyme activity in pH, it has an optimum activity at pH 7 because it's your mouth, right? Pepsin, where is pepsin? In your stomach, right? Your stomach is very acidic, so it has optimal activity in your stomach. Arginase is where? In your small intestine, right? So that's going to have optimal activity in the basic conditions of your small intestine. So that's it's kind of cool. I like that. I think this is really cool that, the optimum, the native form of pepsin is native at pH 2 because that's where it works. That's usually acidic, pH 2, but that's where it works. It works in your stomach where there's very acidic conditions. Do you guys hear about the FDA is now looking at antacids because they contain aspirin? A lot of antacids, like the Alka-Seltzer, some of them contain aspirin in them. And so people don't know that there's aspirin in them. And so they take these antacids because they're having stomach issues. But what they're doing is they're also introducing aspirin, which we know gets absorbed through the stomach through that one homework question, right? And so that's causing stomach bleeding. So the FDA is now saying, okay, these warnings need to be, it needs to be more clearly stated on the packaging that there's aspirin in this so that people don't mistakenly take an Alka-Seltzer product thinking this is just an antacid when it's really got some aspirin in it as well. How do you know that you have stomach bleeding? Do you just have pain or do you?
Maybe like an ulcer?
Is that stomach bleeding is an ulcer? Is that what it is?
It's like breaking your wall.
It's a breaking your wall. So how do you know? Is it just painful or do you throw up blood?
I know some have ones
It burns? It really hurts. You think you're going to die? Yeah, I've never had an ulcer, so. All right, so each enzyme is going to have its own optimum pH. It does not have to be neutral. So this is an important point. All right, I don't really care about this too much, but we're going to classify enzymes according to the type of chemical reaction it catalyzes. So because there's so many enzymes, you have to find some sort of order. So we have a two-part name and a numbering system. However, I don't know anybody that uses this. People use the common names, just like acetic acid is not ethanoic acid. I mean, it is ethanoic acid, but people don't call it ethanoic acid. because everybody calls it acetic acid. Even in the most formal texts that you have out there, they're not going to call it ethanoic acid because nobody does. And that's the same thing for these enzymes. They may have a systematic two-part name. They may have a 4-class numbering system, but no one calls enzymes by their systematic name. It's just not a common thing. So alcohol dehydrogenase. The systematic two-part name is alcohol NAD oxidoreductase. So you can think of that it's a redox reaction that it's catalyzing, and it has a numbering system. No one will walk around calling, if they're publishing in a paper and they've used alcohol dehydrogenase, or they're studying it, they're not going to call it alcohol NAD oxidoreductase. It just doesn't happen. So that's why I'm not going to focus on this. The only thing I want you to notice is what is the ending to all of our enzymes? ACE. All right? That's what I want you to remember from this. It's an ACE and A. But I do want you to know that there's only a few different types of enzymes, right? And these are the different types. So oxidoreductases, they're going to catalyze redox reactions. Transferases, they're going to transfer groups, right? So the naming isn't really that bad, right? Hydrolases add water across a double bond. They cleave bonds, so they hydrolyze by adding water across the bond. So the oxygen is going to be like a nucleophile, and it's going to break a bond. Okay, so this is hydrolase. You're going to break a bond using water. Lyases will form double bonds, or the reverse. They remove groups. Isomerases, isomerize. Mutases, you'll see that. A mutase, that will be the name that we see. And ligases put two things together. They'll often be called synthetases. So just keep this in mind. Okay, I'm not going to have you list the different classifications of enzymes. I'm not going to do that. Just keep these in mind as we go forward, especially when we go through our pathways. Try to think back. Oh, this is a mutase. What does it do? So as we're going through, maybe look at this list as we're going through the pathways and say, oh, that makes sense that this is called that because this is the kind of reaction it catalyzes. All right, so what we're going to do now is do our kinetics review. Remember, again, that thermodynamics is going to tell us if the reaction is going to proceed, negative delta G. But it's not going to tell us about the rate. Our kinetic studies are going to tell us about the rate, and I'm more interested in the mechanism. My background is drug discovery, pharmaceuticals, so what I used kinetics for is to tell me about the mechanism by which a drug would bind to a protein, and kinetic studies can do that because it's going to tell us about the bumps. All right, it's going to tell us about the molecularity of the reaction. So if it's been a while since you've taken Chem 2, pay particular attention to what we're going to be talking about now. If you just took Chem 2, then you should be good. This is a review. You might want to take a look back. If anybody has a difficult time with this, I do have resources in my office that you can take a look at. So if you're having a hard time with this, but I'm going to walk you through it right now. All right, so let's take this reaction. We have two molecules, A and B, reacting to produce a product. We can say the rate of the reaction is given by these general rate equations. So the change in the concentration of A over time. How does A change over time? How does it disappear? Now we have this negative here because it's disappearing. Remember, delta is final minus initial. If it's a reactant, it's going to be going down, right? So if it's final minus initial, then it's going to, delta A is going to be a negative number. But rate is always going to be positive, right? Rate in the four directions is going to be positive. So if we're expressing the rate in terms of a reactant, there's going to be a negative in front of it. because the delta means final minus initial. So we can talk about rate of a reaction by expressing it in the change in concentration over time for any one thing in a reaction, any species in a reaction. But we also have the rate law. And the rate law is going to be more indicative of what's going on with our reaction. So the rate is equal to some constant. Now remember, these brackets mean molar concentration, right? It means molar concentration. So the concentration of each reactant raised to some power, right? And the power is what we call the order of reaction. So we have an order of the reaction with respect to A, and we have an order of the reaction with respect to B. And then K is some sort of proportionality constant. And the only way that we can determine both the K and the order of the reactions is by doing experiments. You cannot look at a balanced reaction and determine what the mechanism, what the rate law is. Now, if we have it first order with A and first order, so F and G are both 1s, then the order of the reaction is going to be 2 overall. So you add the exponents in order to get the order of the overall reaction. So say if F was 1 and G was 1, then we would say that the reaction was first order with respect to A, first order with respect to B, but second order overall. Does that make sense? Coming back to you guys, how long ago was Chem 2? 7 years. It's a while. How long for you, Denon? 3.5? So, is it coming back to you? Yeah? All right, that's good. Okay, we covered it. This is like the first thing we do in Chem 2 here. So it's, even for people that just took it, it's still back in January. So it's a while ago. All right, let's take a look at a different one. So say we have ones. Okay, so I just went over this. I kind of got ahead of myself here. So if we say that we did an experiment, and we'll go over what an experiment would look like, and we found that it was 1 for A and B, and we say it was first order in A, first order in B, and second order overall. So how do we get these? We have to do experiments. And this is what an experiment would look like. You would take a reaction. So this is a dipeptide, 2 glycines together. We're going to add water, and we're going to hydrolyze that diglycine to 2 individual amino acids. And here, this is molar concentration. So this is A, B, and C, 1, 2, and 3 experiments. So in the first reaction, we're going to keep the concentration of diglycine at 0.1 molar, and we're going to keep the concentration of water at 0.1 molar. And then we're going to measure the initial rate of the reaction, the change of the concentration of, maybe we'll measure the concentration of the product. This is pretend. And we measure the rate. And then we change the concentration of one of the reactants, but we hold the other reactant constant. So if we look at, we're holding this one constant, and we've doubled the diglycine concentration. What happened to the rate? That's how it doubled. So if we double the concentration and we double the rate, then we say it's first order. There's a more mathematical way to approach this, but I'm not going to get into it here. Okay? So if we double the rate, or if we double the concentration and the rate doubles, then it's first order. If we double the concentration and the rate quadruples, it's second order. Sure, let me go back. If we double the concentration and the rate doesn't change, it's called 0 order. If you double the concentration and the rate doesn't change, it's 0 order. If we double the concentration and the rate doubles, it's first order. If we double the concentration and the rate quadruples, it's second order. So if it went from 1 to 4 times 10 to the minus 2, we would say that it would be second order with respect to diglycine. Again, there's a more mathematical way, and if you'd like to explore that, we could take some time after class, but I'm not going to muddy that here. What order is it with respect to water? You have to look at the two reactions where the other one is kept constant. So you have to hold the other reactant constant. So the diglycine is in the A and C is held constant, right? So we're going to take a look at the rate, how the rate changes when we double the concentration of water. And what happens to the rate? Doubles. So what is the order with respect to water? First order, right? So when the diglycine is constant and we double the water, A and C, then the rate doubles. When we double the water, or hold the water constant, and we double the diglycine, the rate doubles. So X and Y are both 1. In real life, it doesn't work out this way. You get strange things. It doesn't. So I was in grad school, I was doing experiments like this. But I was looking at the kinetics. So for all of our anti-cancer drug research, we were looking at, we obtained tubulin from cow brain. Right? Everybody did that. It was just very easy to do. We would go to the slaughterhouse and get cow brains and isolate tubulin from the cow brains. And we would do these studies of the effectiveness of these different antimicrotubule drugs with the cow brain. However, we also knew that the amino acid sequencing of tubulin obtained from other parts of the body, cancer cells, the amino acid sequence was different. It was called isotypes. And so one of my big projects for grad school was to take a look at the kinetics of the binding of these anti-cancer drugs to cow brain. And what I then did is I grew up cancer cells, and I isolated tubulin from those cancer cells and looked at the kinetics of the binding to the cancer cells, tubulin. And I found that the kinetics of the binding was different. So these modeling studies that we did for the way that these drugs interacted and the effectiveness of the drugs as antimicrotubule agents, they was flawed because we were using cow brain tubulin. And what we really should have been doing, it was prohibitively hard, but ideally what we should be doing is obtaining, so say we wanted a drug for prostate cancer. So what I did is I obtained prostate cancer cells and grew and grew and grew them, and then isolated the tubulin from them, and not easy, and then did kinetic studies and found that the binding was slightly different. And the kinetic studies told me that was it. I used a DOS program. Do you guys even know what DOS is? DOS is like pre-Microsoft Office. It's like, you know the screen when you first turn on your computer? That's DOS. And so everything looked like that. Doesn't do that anymore? My computer does that.
You're talking about like a black screen?
The black screen. Yeah, that's DOS. That's the underlying, underneath everything. So this is just, I tell my story about my graduate work because kinetics is so powerful. And yeah, that was a lot of fun. So what does this one and this other one mean? What does it mean? What does it tell you about the reaction? Having it be first order means that the rate of the reaction depends proportionally upon the concentration of the substance. So if it's zero order, then it doesn't matter if you raise or lower the concentration. So it's not really in the reaction, right? If it's zero order, it's not really in the reaction. If it's first order, then to me, it means it bumps once. If it's second order, it has to bump twice. So say, for some reason, it was first order with the diglycine and second order with the water. It would mean that water would have to be in 2 collisions, and the diglycine would be in 1 collision for the mechanism of the reaction. Okay, when a reaction is catalyzed by an enzyme, we say that the reactant is the substrate, and the part of the enzyme is bound, it's called the active site. So here we said that the reactant is substrate. Here we have the enzyme, and the part that the substrate is going to go into is going to be an active site. Now, what's very important is that we understand that there's in all of our modeling, that we end up with this enzyme substrate complex before it turns into product. And that, look at this double arrow. This double arrow means that it's reversible, that we can have that substrate come back out without forming a product. It's a reversible reaction. So sometimes, here my hand is the active site, and this is a substrate. It can go in and go back out. Sometimes it takes a couple times in order for it to come in and make that enzyme substrate complex. And that has to do with the affinity of the enzyme for the substrate. How many times does it need to? So basically the equilibrium constant here, if it's lying to the right, to the products, then it has a high affinity, right? If we have a lot of enzyme substrate complex and not a lot of free enzyme, that means that the enzyme wants that substrate, right? If we have a lot more free enzyme, not a lot of substrate in there, it means that there's not a lot of affinity for that substrate. How is that substrate held into that enzyme? We're going to use mostly non-covalent forces. It's going to be attraction of positive to negative, hydrogen bonding, maybe some hydrophobic interactions, van der Waals. And sometimes we do have, in our example, when we're going to take a look at the active site next chapter, we can take a look at, there's going to be a covalent interaction there. So there's 2 models describing how does that substrate come in and form that enzyme substrate complex. There's 2 models. One is a lock and key, that the enzyme has a shape that's exactly complementary to that substrate, that it's going before the substrate even comes by, all right? The enzyme has a shape and a charge distribution that's going to exactly complement that substrate. So that's why it's called a lock and key. Induced fit is more of what reality is, that it comes in, the substrate comes in, and it's sort of like a Venus flytrap. It kind of closes in around it. Right? This is a better model, induced fit. It's a more complicated model, but it is something that's it's more of reality. So this is what they look like. So here's the lock and key. The active site looks, here's the enzyme. There's a substrate here. The active site has a shape that is exactly complementary to the substrate before the substrate even gets there. Right? It looks exactly complementary, like a puzzle. However, in our induced fit model, the active site sort of looks like it, but what's going to happen is that there's going to be a change in conformation in our enzyme that is going to change once that substrate gets close. Right? It's going to change and form around it. Now, once we have that enzyme substrate complex, by either lock and key or induced fit, how does it form product? And why does the product then go away? Well, what happens is the enzyme is going to work to make that substrate look more like the transition state. The reaction is going to be catalyzed. It's going to happen. The product's going to be formed. But look, the product doesn't have as many places where it's going to be attracted to the active site. So then it will be released. And what are we left with? An enzyme that can be used over again, an empty parking spot, all right, for another substrate to come in.